Good afternoon, everyone. The topic for today's class is General Epidemiology. The contents are Introduction, Concept, Definition of Epidemiology, History, Aims, Principles and Tools, Basic Measurements in Epidemiology, Incidence, Prevalence, Classification of Epidemiological Methods. In this, we'll talk about the observational studies, which includes the cross-sectional and longitudinal studies and references. Coming to the introduction, health and disease can be studied in three basic ways: observation of effect on individuals, laboratory experiment, and measuring the distribution in population, that is epidemiology. So epidemiology, the concept, it is derived from the word epidemic, where epi means upon, demos means people, and logos means study. So it is the study of epidemics in human. Definition: it is defined as the study of distribution and determinants of health-related states or events in a specified population, and the application of this study to the prevention and control of the health problems. This definition was given by John M. Last in the year 1988. So this is how we do the epidemiological studies. History: the first known epidemiologist is Hippocrates, and John Snow is known as the father of epidemiology. The components of epidemiology are disease frequency, distribution of disease, and determinants of disease. Principles: there are four principles: exact observation, correct interpretation, rational explanation, and scientific construction. The tools of measurements in epidemiology include rate, ratio, and proportion. Rate: it is the frequency of disease or characteristics expressed per unit size of the population. Further specification will be the time during which the cases have occurred. So different types of rate are crude rate, specific rate, and standardized rate. Ratio: it is the number of persons affected relative to the number of unaffected persons, not relative to the total population. Example, number of children with dental caries divided by the number of children with malnutrition. Proportion: it is the number of cases of a disease is sometimes expressed relative to the total number of all cases of disease rather than to the population, which is expressed as percentage. Coming to the basic measurements in epidemiology, there are two: measurements of mortality and measurements of morbidity. Mortality is the condition of being mortal or susceptible to death, whereas morbidity is any departure from a state of physiological well-being. So for morbidity, we measure the incidence and prevalence. Incidence is the number of new cases occurring in a defined population during a specified period of time. So this is the formula for the incidence. And prevalence is the estimate of the proportion of individuals in the population with a given disease, disability, or the health state at a particular point in time, which can be of two types: point prevalence and period prevalence. So it includes the current cases, both old plus new. So this is the diagram showing the relationship between the incidence and prevalence. So incidence is the new cases, whereas prevalence includes both the cases. Next, coming to the classification of the epidemiological methods. The epidemiological study designs are basically classified into two categories: the observational studies, which include descriptive and analytical. In descriptive, we have cross-sectional and longitudinal, and in analytical, case control and cohort studies. And in the experimental studies, we have randomized control trials, field trials, and community trials. First is the observational studies, which includes descriptive epidemiology. They are the first phase of an epidemiological investigation. They are defined as the disease concerned with observing the distribution of disease or health-related events in a population in which the disease in question seems to be associated. The steps involved in the descriptive studies are 6, which we will talk in detail. So first is defining the population. Defined population may be the whole population or representative sample of that population. It can also be specifically selected group like age and gender groups, occupational groups, hospital patients, school children, community, etc. Second is defining the disease under study. The epidemiologists look at an observational definition of disease in question, a definition by which the disease can be identified. Definition may not be precise as that of physician, but adequate enough to identify the sufficient accuracy. So this is how we define the disease under study. Third is describing the disease. The disease is examined by the epidemiologist by asking three questions. When is the disease occurring? Time. Where is it occurring? The place. And who is getting the disease? Person. So we describe the disease in terms of person, place, and time. So time can be year, season, month, places, the person on the basis of the various characteristics. Coming to the time distribution, it yields important clues about the source of etiology of the disease. As we can see three type of the trends in the time, which includes the short-term fluctuations, the periodic fluctuations, and the long-term fluctuations. The short-term fluctuations, the best known are the epidemics. So epidemic can be defined as the occurrence in a community or the region of cases of an illness, all the health-related states or events clearly in excess of the normal expectancy. So whenever a case is more than normal, it is known as the epidemic. So these are the various types of the epidemic: propagated, seasonal, cyclic, common source, point source, etc. Second is the periodic fluctuations, which include seasonal and cyclic. The seasonal variation is well-known characteristic of many infections like measles, upper respiratory tract infections in winters, etc. And cyclic fluctuations, which occurs in cycles of short period. Example, measles once in 2 to 3 years before immunization. So this comes under periodic fluctuations. Coming to the long-term or secular trends, it implies the changes in the occurrence of disease over a long period of the time, like coronary heart disease, lung disease, diabetes, etc. Second is place distribution, which includes the geographic differences in the disease prevalence, which are important. It includes international variations, national variations, urban, rural, and local distributions. Third is coming to the person distribution, which includes the study of the various host factors in relation to the occurrence of the disease. So it can be age. It is an important host factor strongly related to disease occurrence. Example, measles is common in children, cancer in middle age, and degenerative diseases in old age. Gender, the gender ratio, gender-specific mortality and morbidity rates, male-female ratio in the prevalence of lung cancer to identify smoking as a risk factor, and variations related to the biological differences like genetic inheritance, cultural differences, etc. Third is race. Differences in the disease occurrence have been noticed among the population of the different ethnic origin like the sickle cell anemia, hypertension, etc. And other host factors includes marital status, occupation, social class, behavior, stress, migration, etc. Fourth step of the descriptive epidemiology is the measurement of disease, which includes cross-sectional studies. It is the simplest form of the study based on single examination of a cross-section of the population at one point of time, also known as the prevalence study. If the sampling methodology is accurate, results can be projected, and they're very useful for the chronic diseases. Next is the longitudinal studies, which involves repeated observations on the same populations over a period of time. So it measures the incidence of the disease in which there is a long period of the follow-up of the patients has been done. Fifth step is the comparison with the known indices. By computing various rate, ratio, and proportion and making comparison, it is possible to arrive to the clues of the etiology of the disease. And last step is the formulation of hypothesis, in which hypothesis is the supposition arrived from the reflection or observation, which can be accepted or rejected using techniques of analytical epidemiology. So success of research project depends upon the soundness of hypothesis. Example is smoking 30 to 40 cigarettes may cause lung cancer in 20% of smokers after 20 years of exposure. Uses of descriptive epidemiology is to provide data regarding types of disease problems, their magnitude in the community, and to provide clues to the disease etiology and help in the formulation of the etiological hypothesis. Third, to provide background data for planning, organizing, and evaluating preventive and curative services. They contribute to the research by describing the variations in the disease occurrence by time, place, and person. So these are the textbooks which can be referred for the further reading. Thank you.